Okay, so in the previous session, we talked about some basic skills on single cell data analysis using one sample in Partek flow. And today, we're going to talk about how to analyze multi-samples of single cell RNA-seq and what are the options that you can analyze the data and different ways to visualize the data. And when you have a lot of samples, so sometimes you might have a batch effect. We're going to cover how to do the batch removal and also cell type classification, differential gene expression detection and between different cell types or between different phenotype, etc., different options. And also when you have multiple samples, you might not only look at the differentially expressed genes, you might want to look at the difference of the cell number in between different phenotype. We call that a cell type abundance analysis. And also, if you have replicates at a sample level, and a different way to look at your data, we call that a pseudobulk analysis. Also, I will introduce you how to do automatic cell type classification. Of course, this rely on the database. So you have the restrictions on what kind of database you can find. After the single cell RNA-seq analysis, I will talk about the site-seq data analysis, the options in Partek flow, and then Visium data analysis to show you how to analyze the spatial transcriptome data. All right. Okay, so today is the example data for single cell RNA-seq I'm using is download from Geo. And this data set, there are 12 samples. It's the human PBMC data. Within the 12 samples, there are two different age groups. There are 5 samples. They're young. Actually, they're not really young. They are between 80 to 100, 50 to 80 years old. And then super centenarial, that means those people, they live for more than 110 years old. And there's male and females. So the whole point of this, the design for this study is to study the age group. All right, so now in the analysis tab, you're going to do the same thing. After you import the data, you're going to do the QA/QC and use the same concept to clean up the data to filter out you think it's not viable cells and filter out low expression genes. And also in Partek, if you click on the task, you can collapse a pipeline if it's too long. So I try to collapse it to focus on after we got normalization. Okay, so this is the normalized data in the logs in the log base. So the first thing, last time when we talked about to classify the cell types, we always want to visualize the data, right? And then when we visualize the data, we rely on dimension reduction, like a PCA, UMAP, t-SNE, etc. Okay, here, I also want to talk about that when you have multiple samples, people always think, okay, do I have a batch effect? This is my thought about batch. When you see batch, usually that means it's non-biological factors will cause the change of the genes of your data, right? So for example, if my data is processed by different labs or by different prep kits, and then that might cause some batch. Or if I want to integrate my data with the data downloaded from Geo, and then you might have a batch effect. So however, I wouldn't recommend you to remove the batch right off the bat because first of all, if there's no batch effect, you can't, if any batch remover is going to change your data, you can get closer to the truth or you might get further from the truth. You don't know. So I would like to look at the data first before I decide that there's a big batch effect. For example, when I look at visualize the data in all kinds of visualizations, I do see all my data is on the left side and all the other data is on the other side, and the batch effect is much, much bigger than the biology effect. And then I will do another iteration to remove the batch. So remember, data analysis is iterative, especially for single cell data. So when you try to remove the batch effect, this is really depends on your experiment design. Within each batch, you should have your biological replicate. Otherwise, if your biological effect and the batch effect is confounded, then you're stuck. For example, I saw some users, they have two samples. One is treated, one is control. And after they import the data, they say, oh, I want to remove the batch to do like an integration. That can be very dangerous. If you remove the batch, which means the difference between the two samples, you're going to remove your treatment effect. Okay, so in that case, it's going to be very hard. I couldn't tell there's a magic algorithm can only remove the technical variation, but keep the biological variation. So that case, I would say don't remove the batch effect. Usually, if you only analyze your data and your data processed in one lab or in one, I would say one batch, you really don't need to worry too much batch effect. Only when you try to integrate your data with other people's data, or if you integrate the data from different assays, like some samples processed using one prep kit, the other one using a new version of the prep kit, and then you might have a batch effect. In that case, yes, it's reasonable to remove batch effect. I still recommend you to analyze your data. The first iteration, just pretend they don't have batch effect until you really see a clear batch effect. Batch variation is much bigger than your biology variation. And then you come back, remove the batch. So within Partek, in terms of the batch removal, we do have several options. One is after you're normalized on the normalized data. So this data node contains the matrix. Every cells with every genes is normalized count. On this data node, if you go to batch removal, we have two options. One is a general linear model, which is based on ANOVA. This is a Partek way to remove the batch. So it's almost like a batch ANOVA reverse. So the algorithm is using ANOVA. And the other one is Seurat3 integration. So Seurat3 integration, once you do this, it is really helpful for you to do the downstream visualization. So after you use a Seurat3 interaction, I would recommend you to, just use the visualization. After PCA, you can perform a cluster and perform visualizations, identify your cell type. So visually, you wouldn't see the batch effect. And then you're going to use the previous technique we learned to classify your cell type. However, if you say, I want to remove the batch effect to detect differential expression genes, in that case, I wouldn't recommend you to use batch remove the data to do it. Actually, you can go back to the normalized count. Whenever you use differential statistics, most of the models like DEC, Hurdle model, and ANOVA, all those models which can take multiple factors in the model, you can just add your batch in addition to your biology fact. So suppose I want to compare the cell type and I want to take the remove the batch, just add a cell type and a batch, both factors into the model. So the algorithm will partition which variation coming from the batch, which variation come from your biology effect. It will take care of that. But only some other algorithm, like those algorithm, like non-parametric analysis, they won't. You can only add one factor. So in other words, some statistical method can take care of the batch removal. So you can use batch only for visualization. And another one is really pure for visualization is you can, when you click on the PCA data node, PCA only contains PCA scores. It's basically for visualization. And if you go to the batch removal, And here is another way called, it's in public domain, pretty popular way called a harmony. That way, you can look at your data, visualize your data before batch, and then use harmony to remove that, and then perform UMAP to look at, after batch removal, how the data look like. So I would highly recommend use the batch removal. It's really just for visualization purpose. When you perform a differential analysis, you're going to come back to your data node and use a statistical method to remove the batch effect. Okay, that's my recommendation. However, like what I said, if you only have 2 samples and you really don't know the difference between the 2 samples, is really biology effect or is really a technical issue caused the difference. If that's the case, and a Partek in the visualization, we have another way. It's not a batch removal. I think you can ignore your batch effect for when you classify your cell type. When you visualize your data, if you have a big batch effect, the batch effect kind of hides you won't be able to discover the biology visually, biology effect. So if that's the case, when you perform PCA on this data node, when you perform exploratory analysis to PCA, we have this option, split by sample. So whenever you do, when you have more than 1 sample, Partek will give you this option. So I would highly recommend you to perform both ways. You're going to first to perform PCA on each sample individually, but only with 1 button click. And then you uncheck it, you can combine them together. I can show you how to use that. So this is what I did. After I do PCA, I would highly recommend you rename the PCA. So that way, okay, this PCA is on each individual sample. And on this one, when you perform the graph-based clustering, it will also perform each sample individually. So, and also when you do UMAP, TSNE, you're going to look at each sample separately. And when you look at the graph-based clustering, graph-based results, you will see every sample, how many clusters I have. Okay, and then when you click on each sample, again, you will have the details of, which clusters have how many cells and what the percentage, just like when you perform 1 sample at a time. Okay. Now let's look at, I'm still going to use UMAP. You can double click on the UMAP to open this visualization in the current browser tab, or you can open in a different tab. Now this UMAP is only look at 1 sample at a time. So you can pretend I only have this 1 sample. Sorry, since I already classified the cell type, and you probably initially don't have anything, right? Just like that. So you look at your data, just like visually, I can see different clusters. And then in the meantime, if I want to use a graph-based information to help me to identify the cell type, and I can go to get data and search for the graph-based data node. And then when you do the graph-based, you'd better label them nicely. And this one is for each individual one, and you can click on it and go to the graph-based annotation and color it. So graph-based clustering partition this particular sample into 6 groups. And you're going to see 2 numbers. The first number is the unique ID of the sample. The second number is the cluster of this sample. And just like what we learned in the previous session, I can look at the biomarkers. I'm going to go to the get data and search for the biomarker table. I can put it over here. Just like you only have 1 sample And then you classify the cell type. For example, what we learned here, I like to, look at both side by side. One is using statistics to partition the data, to color the data, and the other one use the biology, use the gene expression to color the data. So I can clearly see, oh, this look at the T cell. And also you can use the select and the filter, and by using the gene expression to select the cells. So the techniques on how to select cells and give a name of the biological meaning name of the cell type is the same as we learned in the last time. Okay, so I'm gonna just suppose I want to select, CD3D is greater than 7. I will call that T cell. So now I select 1,981 cells. So all those cells, I will call that classify. I call that T cells. Okay. And you're gonna do the same thing, go on and on. For example, let's see, cluster 4, see, because I see CD79A, cluster 4 is the green color. So probably this is B cells. Okay, now this time I can manually call that, classify, call that B cells. Some of them, they already classify in the T cells, but the later label will override the previous one. And you're gonna keep on going and finish all the cell type you can identify it for this sample. Okay, so now I want to go to the next sample. What can I do? You can select the top, those two, they're the same graph, and then you go to the axes, and on the right side here allows you to look at other samples. You can use the drop down to go to the next sample, or you can just go to the next sample. But this one, since it's a table, they don't have the axes. So you go to the content and then go to the next sample. So now we're looking at sample 2. Oh, this one somehow. Okay, make sure this is sample 2. And then, you use the same technique and identify, your T cell, B cells. Okay, so suppose I want to, I can clearly see this group of the cells, I think is T cells, okay, because the CD3D is high expression. And at this time, I'll just select this cells. Then you go to classify. And then when you click a classify selection, do not give a new label. Still call it T cell. T cell is a T cell. It doesn't matter which sample. Just think about this concept. Watch this number. Previously, I have a 1972. Now I have a 1637. When you click save, Partek will automatically combine the cell type from different sample. So this is another way for you to look at each sample individually. The advantage of this way is when you classify each sample, focus on one sample. First of all, you don't need to worry about the batch effect. And second of all, because the cell number is small, and you will have a higher resolution when you visualize your data. So let me see if I use maybe this one. Just color that. Okay, so this is B cells, right? Call this. Again, just call B cells. So do not call that a sample 1 B cell, sample 2 B cell. Do not do that because Partek already know which cells are from which sample from the data tab. Okay, just call the same thing. One trick is you probably won't be able to finish the whole project within 1 day. Always save it. Say, okay, I'm gonna save this session. You can save as, suppose I will call that analysis in process or something like that. So now you log off Partek, and the next day when you go back to the data viewer and go to where you left off and continue analyze your sample 3, etc. Okay, so now you still where you were when you go to the classify, and that's what you classified all the cells, and it's gonna keep on going. After you finish all your samples, do not click apply classification if you didn't finish the first iteration. So you can always save the session. But once you finish everything, and that's all I can identify, you probably still have some cells you won't be able to, but that's fine. This is my first iteration. Once you finish all the samples, all the cells you can classify, you click apply classification and give a name. Call that, my cell type version 1 or something. And once you click run, the information will be saved. If you go to the metadata, the information will be saved at the cell attribute. And you can see it's right here. Okay. So you imagine in a spreadsheet, each row is a barcode. And every time when you classify the cells, and this is just like one column, and you want to assign the information of your cell type for ideally all your barcode, right? So you don't want to classify half of your data, you apply that, then the other half of your cell type, they're going to be NA. And you're going to have a hard time to merge 2 columns. So this is only 1 column, and then you have each barcode. You have the cell type. You give assignment to it. And previously, I already have the 4 cell type classified. So this is the technique I really highly recommend you to use this unique things in Partek flow to analyze each sample individually. But if you have a lot of samples, so suppose I have 100 samples, and my major classification technique I want to use is based on gene expression. If that is the case, you really probably don't need to do this way. You can, when you do cell PCA, you can choose and check the split sample. So this PCA is on all the samples together. And then if I look at this UMAP, and a UMAP or a graph base, you can look at this. So there's 8 clusters for all my samples. Okay. So now if I look at this UMAP, again, this is I already classified the cell type, and that's all the samples. And the nice thing about this one is you can, look at all samples together. And also within this type, there is the option, there's a grouping option. You can choose a split by sample. So now this is all my 12 sample, but they are in the same coordinates. If I rotate them, they all rotate together. The nice thing about this visualization is I can eyeball the same location to see all my cell types. For example, I can clearly see there's more cells over here in this sample compared to this sample, right? So you can do the like a cell number comparison, just eyeball it, just visualize it. Okay. Now I want to show you a technique is how can I combine, both UMAP, one is individual one and one is all together. Let me just do an undo here. So this is the UMAP of all the samples. And in the same time, I want to look at each individual sample. Again, you go to get data and search for UMAP. And that one is invoked from UMAP all. And this one is I'm going to use the UMAP individual. I can drag this data node, use the information from this data node to make a plot. I will make it on the right. And I can do a 2D or 3D. Okay, let me just do a 2D. This is a sample one in the sample one only in the UMAP. And you can see the location will be different. And if you want to select, if you want to say, how about sample two, use the duplicate technique, sample two, and this one, go to axes, change to sample two. And you can build your 12 samples this way too, but the coordinate is going to be different. You cannot look at the, one place. So sample one and sample two, they are going to be in completely different space, different coordinate, but you will have a higher resolution. And this one, if I select, sample ones, this group of cells, they will be highlighted. If you look at all the samples, they are right here, right? That's why I said when you look at each sample, you can clearly see the separation. But when you look at them together, there's some other samples, certain cell type, probably, over here, you have a lower resolution. And then this one, you can split. So those are the different ways, whether you look at one sample or all samples together, different ways, different perspective to look at your data. They all have a pros and a cons and depends on how you use them. Okay. So that's the one technique you can utilize when you analyze multiple samples for cell type classification in Partek. Okay, so now I have all the cell type classified in the data. So the next thing is your goal is to compare old versus young, right? When you design this experiment, you probably want to compare that. When you do bulk RNA-seq, all the T cell and B cell, they all clustered together, they all, kind of pulled together. You really don't know. Some signals might be canceled. Maybe this gene in the old people will showing upregulated in B cells, but downregulated in T cells. But if you use a bulk RNA, you won't be able to do that. So that's why single cell give you a high resolution to look at each individual cell type, and then you can compare the age. Okay, so one way to compare the age is, when you mouse over this normalized count, I have 12 samples, so 57,000 cells, 7,000 features here. One way is it's easier to do. You can use this function. So when you detect a differential analysis, if you use a Partek default normalization, because you're still going to have zeros, you can use a hurdle model or ANOVA. So both the method is what I recommended. If you use other, like some other tools normalize data, depends on the distribution, you can choose a different, lemma or DE-seq. I wouldn't recommend you to use the DE-seq on single cell data. So those two options is what I recommend. And then you can, typically you're going to include your phenotype information, age, and your cell type information. So you can add those two effect into your model, and also you can add interaction. So when you select more than 2 factors, you can add interaction. So add interaction will give you options to answer more questions. So you can compare young versus old, or you can compare different cell type, or the other one is you can compare, somehow my internet is kind of slow today. Step is slow. Okay, so you can compare, suppose I want to look at the B cells, young, and the B cells, old, right? So this is the interaction term will give you this option. Or you can compare, somehow it's really slow. Oh, okay, now finally I got it. Move this. Okay, so you can ask all the combination, or within the young people, I want to look at the B cells versus T cells. Okay, So typically after the cell type classification, all the downstream analysis, all the comparisons is very much the same as you analyze any RNA-seq data. So this is the option you can do. But I really don't recommend you to do it this way, because even you compare B cells, young versus old, your other cell type is still in the calculation to calculate the variation. That is ANOVA. It's not a perform t-test. What I really recommend you is you can click on the normalized data node and go to pre-analysis tool, go to filter, I'm sorry. You can filter only a certain group. So you can choose to filter cells, only filter a certain group. Or another way, the split by attribute is kind of automatic filter. So if you choose to split by attribute, you will see I want to split based on my cell type. And you can generate automatically. Partek will perform 5 filters, and there's some cells I haven't identified as called NA. Or if you uncheck that, I want to perform 4 filters. So it's like a more advanced filter. And after you've done that, that will give you the different data node. So this data node, if you mouse over, it will tell you you still have your 12 samples, and there are only B cells. It's 4,000 B cells. And within that, it's almost like you purified your cells. Now I only focus on B cells. I can perform a differential analysis, compare old versus young, and also I can perform pathway analysis, perform GSEA, or all the downstream analysis. And you can do it on different cell type. So once your cell type is classified, you can separate each cell type and to compare your phenotype to look for genes showing differential expression. Okay, so that's typically, most people will ask this question. Another thing is on top of that, especially when you have a multiple replicates in each group, if you only have one sample versus the other, or only have one sample, you probably won't ask this question. It's called the cell type abundance analysis. What that means, let me just go back to the data visualization. I want to show you, I will start from my normalized count, because when you identify the cell type, put it in the cell type attribute, that tab, that means this attribute can be seen by all the data node. So I will always use my normalized count as my start point. I will use this data node. I want to make a bar chart. And the bar chart, I can separate them. Each bar represents, suppose, a sample. Okay. And this is the y-axis is how many cells in each sample. This is not very useful. But now if I want to color, go to style, I want to color by cell type. And this is each sample, the composition of my cell type. And this is the absolutes. Some samples you have more cells, some you have a less number of cells. If you want to make them the same, say, I want to look at the proportion, you can choose 100%. So now you will see it's kind of the proportion. Based on this, I can clearly see those two samples. The blue colors B cells, they have really few number of B cells in those two samples compared to here. Right? So this is you look at the composition, cell type composition in each sample, or you can visually look at each group. So suppose the x-axis, I can change that to age. And overall, I want to compare young versus old. I can clearly see in the older people, they have relatively less number of cells, and instead they have more T cells over here. Okay, and also you can add gender if you want. If you want to add both, you can choose a categorical list and say, okay, one category is age, the other category is gender. You can do it this way. Okay, and look at this is a young male versus female, and this is old male versus female. And also from here, you can switch the order. So it depends on which graph will help you to tell your story. So all those visualizations, one is to help you understand the data. The other one is to help you efficiently deliver your discovery, your message, your presentation, right? How you want to present the user, tell your story. Actually, to visualize the cell type composition, another way is a pie chart. It's the same thing. I can get the normalized data node. You just choose a pie chart. And then maybe I want to look at each pie represent a cell type. So now this is overall, there's more T cells compared to B cells. But in the meantime, you go to axes, you can add another one. So, okay, I want to add age. And you can switch the order. Whatever you think is clear to for you to help you to tell the story. And also in the meantime, I can split by gender. So this is a male, this is a female, how the composition look like. Well, every time when you mouse over, you will see the percentage, you will see the total number in this section. Okay, I think a bar chart and pie chart is very nicely to visually look at the cell type composition. However, if you say, I want to perform statistic test, do I really see the B cell, they have a less number of B cells in the older people versus young people. Now, if I go back to my analysis, we have a way. So first I filter out the unknown. So this one just filter out the cell type is unknown. And then on this one, you can choose in the pre-analysis tools. We have a task called generate cell group cell count. You click on that to see, okay, what cell number you want to generate. This is performed on each sample individually. So you can look at, my cell type, choose that and click finish. The result will give you a table. You can download this table. You can see, okay, every sample, this is all my 4 cell types. That's the absolute number of the cell type in each sample. And then in the optional column, you can add, those informations here and just eyeball it. Suppose I want to sort based on B cells. You can see those are the older people. It's kind of on the top. Just eyeball it, I can see overall older people, they have a less number of B cells, right? Now, if I want to perform statistical tests to see if this is, since you have a replicate, if you want to perform statistical tests at a sample level, you have to have a replicate. If you only have one sample, then you cannot, right? But when you compare the cell number and you will say, okay, what if this sample, I sequence more cells than the other. So you definitely want to normalize that. And proportion usually is a good way to look at the percentage, right? So in Partek, to normalize your count data to percentage, how do you do that? You go to normalize, normalization and scaling, normalize, and basically for each sample, percentage is you divide by the total number of the cells for the sample and then multiply by 100, right? So if you know the equation, you will say, I want to divide it by the sum for the sample. You cannot perform on the feature, okay? So for every sample, divided by the sum and then multiply by 100. And click finish, the result will give you the percentage. Okay, you can you can look at for example, PCA of this. This is at a sample level now. It's not a single cell because you look at the number of each sample, and based on PCA, you might see there's some separation. It depends on what angle. This angle I can see the separation of young versus old, and then depends on what statistic test you want to use. Since this is a percentage, I can use non-parametric analysis. Here, I just use the cross-Wallace to compare young versus old. And the result is each row is each cell type. And I compare young versus old, the cell type, percentage, cell number. I can clearly see the p-value of B cells is significant if you use a 0.5 as a cutoff, right? And the fold change, young versus old, young, it has more than old. At this time, you can also do a dot plot. The dot plot will be at the sample level. I can see, okay, my each dot is a sample, and the y-axis is a B cell percentage of this sample. Over here, again, we learned you can do a box whisker or violin, and we have a few number of samples. The violin is not that nice. I can, overlay that and color it. And in the meantime, if you say, oh, how about the other cell types? Again, I like to use the duplicate plot. I can put it on the right, double click on the y-axis. I can choose, monocyte. And as you can see visually, they don't see, especially, the box whisker, the median is different, but we see they have a more spread out. And you can look at the other one. And this one, I look at the T cells, right? Or NK cells, etc. So visually, you're going to present your results to say, hey, older people, they have a less number of B cells. That's called the cell type abundance analysis. We have multiple samples, so we can answer this type of question. Another thing is, once you identify your cell type, at the very end, if you want to present, okay, I do have this marker gene, so they can really differentiate all the cell type. So you discovered your marker genes, and you want to present, those genes can really differential, different group, etc. So suppose you have the genes of interest, and you want to present the genes that to show people they can, differentiate whatever, how you generate this, the genes, the different groups. I want to show you heat map. Is a nice way to look at multiple genes. Suppose here I got 4 genes. I have a CD3D, CD79A genes. I know those are marker genes for T cells and B cells, etc. So I got my gene list, and you're going to use the gene, here is add this gene list here. I think this is my marker genes. And I just want to present those 4 genes and to say they are the marker genes for my 4 different cell type. Okay. So now, one way to present it is to, to use a heat map. Again, the heat map, I want to plot on the normalized count. And it's the same thing. You can use your differential expression genes to look at the old versus young. Okay, so I want to use this data node and to display those 4 genes at the cell level. You go to exploratory analysis and hierarchical clustering. You can choose to order them, but at the cell level, I recommend you want to manually order them. So I won't perform a cluster. Instead, I want to assign the order. And the order I will use is my marker gene. So if you have a gene list and you have a certain order of the genes, suppose, you order them based on a certain pathway, make sure you make a gene list. And what happening is that in that data node, it contains 7,000 genes, and it will find the overlap of your gene list over here, filter down to this gene list, and also use the order of this gene list. Okay. And also the cells too, I want to order them based on my cell type. Because a single cell, you're going to have a lot of zeros, very noisy. So you definitely want to group them in some meaningful way. And also you can filter them, and in the data node, there's also NA cell. I couldn't identify them, and you can choose to, filter them out. Otherwise, it won't look pretty. So I will use my cell type to exclude if the cell type is NA. Okay, and do this configuration, click finish, and the result will be like this. This is my 4 genes, and this is each row is a cell. So you look at the cell level, but I group them at the cell type. That's why it looks nicer. And if you say, I don't want to look at the barcode here, you go to axes. We can turn off the barcode. And instead, I want to put the annotation. You can add your cell type annotation. And if you say, I want to make genes the other way, on the right side, there's a transpose button. Some people might look at it this way. And if your genes are really marker genes, I think this heat map is really compelling. For example, CD78A, I can clearly see this is marker gene for all the cells. Every vertical or column is a cell, and in the group of the cells in B cells, they really have high expression. And this one is a CD14, and then this is NK, and this is, well, NK gene marker genes probably is not that great. I can clearly see, this NKG7 have a high expression also in T cells, which kind of makes sense, right? And this is the CD3D is high expression of T cells. And also you can, if you didn't use a cluster to order the cells or the genes, you can change the order yourself after you perform the heat map. So in the vertical mode bar, there's a reorder mode, this icon. You click on that, now you are in the reorder mode. Suppose I want to make this gene above NKG7, you mouse over, or it's going to be green, and green can be draggable. Click on it, and you can, swap those 2 genes. So it depends on what you think is pretty, and you can present the result. Okay, so this is one way to present your marker genes and to tell the discovery those genes can differentiate the different cell type. Another nice way is using, we call that a bubble map. Instead of we look at each individual cells, I want to group up the cells into my cell type group. So now, if I go to hierarchical clustering, we call that a bubble map. Bubble map means you want to group the cells based on some categorical attribute information. So we will give you the option how you want to group the cells. Okay, I want to choose group them by cell type. And what statistic you want to group the cells for each gene. I want to use the mean or median. You can use other options, but mean or median probably is more reasonable to do. You can try other options. And in the meantime, I also want to calculate another statistics, because mean is only one perspective. If you have an outlier, your mean can be really off, right? The other options is I want to calculate, think about the marker genes. So suppose B cell marker genes. This gene should have high expression in B cells, and also most of the B cells, they should have a high expression. It's not like I only have high expression. You might have one cell as an outlier, make the mean really high, but all the other cells, they don't have expression of the gene. So that's not the marker genes, right? So the other perspective is I want to calculate the percent of the cells in each group if the value is greater than such and such. If you're using Partek normalization method and your 0 is still going to be 0, so I will just say no expression is 0. If you have non-zero, that means this gene have expression. But if you use any other normalization method, you have to be careful. You have to look at the distribution of your normalized data. Like if you use Sarras SC transform, you might have negative value. So again, you determine what you think is called expression, right? Greater than certain value, I will call that expression. And then I want to calculate the percentage of the cells they have the expression of this gene. So that's the part of we do the group calculation. And again, you want to do the same thing. The order, I still want to use my 4 marker genes and the order of the cell group. You do the same thing and filter, etc. So click finish. The result will be, the reason we call it a bubble map, because by default, we use a circle, and then the color represent the mean value of the group of this gene, the group of cell of the gene, and the size represent the percentage of the gene, of the cells have expression of this gene. So that's why if you look at like a bubble, we call that a bubble map. Okay. Again, you can switch the order using this reorder function. Look at the way you like. So typically you want to look for, you want to highlight the genes if they are more red, means high expression, and also big. That means they are marker genes. Blue means they have a low expression. Small means that they have a smaller proportion of the cells they have expression of the data. You go to the heat map here. The default, the shape is a bubble map. And then the size, you can change the higher expression. We have a higher and low expression. I have another, you can change the value. The lowest cell size is, suppose I use a 10 versus a 30. And so you can clearly see things here. Okay. Yeah, so this is another way. I would recommend you, if you have, suppose 20 or 30 genes with a whole lot more cell types, bubble map is more effective. But if you don't have like 4 genes with the 4 cell type, maybe heat map is nicer. Either way. And also, if you have a lot of genes or a lot of cells, depends on how you fit the screen. Your screen is a rectangle, and when we draw a circle, it's kind of a square. You might not have enough room to have enough resolution to see the difference between small and big. Another way is, not other software to do that, you can use rectangle. I think that will utilize your screen dimension much better. It's the same thing, but because no other people not using this function, because other software, they don't have this function, it doesn't mean you cannot use this to present your result. Whatever you think will help you to tell the story, just use it. Okay, so this is, of course, these techniques can be utilized on, if you only have one sample for the visualization. So now we talked about multiple samples, classify cells using the different techniques, and after you classify cells, what you can do. Also, I got a lot of users want to do automatic cell type classification. So you have to think about that. There's no magic. Whenever you use automatic cell type classification, there must be something, somebody else already built a database for you, and you can use other people's database to classify. Currently, what we have, the cell type classification built into Partek is under classification. The method is called Garnet. And if you want to get the information, you go to the documentation, search for classification, that chapter. And some details you can look at in this chapter, but at the bottom, this is, the Garnet, the classifier they have. Okay, so we build the classifier. They have a classifier. This is not a human readable. This is an R object, and the other one, the marker list, is a human readable. You can download that. Just to show you, this is, and this documentation is talking about. Suppose I don't have a classifier. I want to build a classifier. I know some biomarkers. You can go to some different database to build your own classifier, and you can make a text file just like this. The first row is your cell type, and the second row is your gene name. The format, you have to match exactly the same format. Okay, just read this documentation to build your classifier. Now I'm going to show you, if you already have a classifier, if you go to your settings, library file management, currently they only distribute human and human mouse, and also there's C. elegans. Some certain tissues cell type. It's not all of them. Human, they have more, but mouse, they have less number. So you can go to classifier and click the add classifier file, and you can download. And this is what they distribute. For human, you can see there's a lot. For mouse, I think they only have a brain and a lung, or you can build your own. Okay, so I already downloaded a human PBMC classifier, and then when you classify your cell type, do not use normalized data, because they probably, under the hood, that they have to match with their database. So use the raw count, perform a raw count, and then just go here, classify cell type. And then just choose your classifier, click finish. So it's pretty simple. And the result actually is only that's the different one. This one. The result actually is only, in their database, they only define those cell type, and it will tell you which cells, how many cell numbers. So basically, you think about that, this will only add one column of your data in your spreadsheet. Each row is a barcode. It just add one annotation. If you want to make this annotation to be able to seen by all the different data nodes, what you're going to do is you click on this data node, you choose publish classifier. So you will say, I want to, my internet is really slow. I just take some time to pull up you just give a name of your classifier, and then after you publish, the result will be in the project level. I say this is what I initially published, the Garnet annotation. So once the information is in that in the attribute place, all the data nodes can be able to see that. You can use your UMAP to color by that information. Okay, and then you can use your normalized count to compare differential expression based on this classifier. So another situation is I know the marker genes, but I don't have a classifier. So you first have to you can use your data to train your markers and to build a classifier. So you're going to click on train classifier. And this one will ask you to give you the marker file. That's a text file. You specify your text file and then click finish. So they just use your data and to look for your marker genes and try to identify, to build the classifier. And this result will be like this. On the top is kind of a graph, and the x-axis is ambiguity from 0 to 1. 1 means bad, Small means good. It's less ambiguous, right? And then on the y-axis is all the marker genes you defined in your text file. And those marker genes on the right side is what cell type you defined. So I think the nice thing about this one is you can tweak your marker genes. So some genes you say, okay, this gene, this paper says a marker gene for this cell type, but the other one say not. Based on your data, and after you train it, you can see, oh, okay, those genes are kind of good marker genes for this cell type. But if you see some genes on the right, you define those genes as a marker gene of dendritic cells, but it seems like it's not very good because they have a high overlap with another cell type. And maybe you can tweak your marker genes and then rerun it. So this is the one thing when you use the train classifier to tweak your markers, to identify your biomarkers. And after this is done, you have this train classifier. You can publish a classifier and use it for a different project, or you can go to your data node to say, hey, I want to classify my cell type. And I don't have a managed classifier in my library, but I want to use the project. I just built one. You're going to select this data node. This is the only data node you can use. So you don't have to publish this classifier if you only want to apply on your data. And then that will perform cell type classification and give you the result, which cell is which cell type you defined. Okay, so that's the automatic cell type classification. And right now, there's so many different algorithms out there to do automatic cell type classification. We are also on the call, and currently we're investigating some other algorithms. So you expect later on, we have more options for you to do cell type classification. Another things I want to show you that is, when you use a single cell technology, there is pros and cons. And the con is you're going to have a lot of dropout, right? You have a lot of observations, but you're going to have a lot of dropout, a lot of genes. They don't express in a certain, they are zeros in a certain cells. It's not because they express zero, it's because your sequence depth is not high enough to sequence this gene because they are all random, right? So the bandwidth is not that great. Maybe in the future, you can, it's less expensive. You can filter, you can sequence more. This problem will be less, it's not less problematic, but currently dropout is a problem. So we have a way called a pseudobulk. What we means is you can pull all the cells of the same type back to the sample level. So that way, you can look at the sample level of your data, but this is not a real sample, it's a pseudobulk. It's almost like I purified all the, B cells, and I want to look at the B cells at 12 sample level, okay? And then I can look at the sample level of the data. So this is what we call the pseudobulk analysis. I would recommend you to use the raw count instead of normalized count, because if I want to pull cell back to the sample level, I want to add them up. It's almost like I will sequence more on this sample, right? Or if you use normalized data, it depends on your normalization. You might want to use a different statistics. So this is what my recommendation on the raw count. And then you choose pseudobulk under pre-analysis tools. Choose the pseudobulk. And then basically it's aggregate cells into some kind of a group, and you define what the group that is. You can use multiple ways to identify. So for example, I just want to look at each sample. So if you do it this way, and then I will use the sum. So what's happening is that it's almost like I just pull the data back to the bulk level and put all the different cell type into my sample. I want to look at how at a sample level the data look like. Since I already identified the different cell type, what I want to do is, not only look at the sample, I can look at the cell type level within each sample. So I look at both, which means I look at the B cells in sample one. Suppose I have 4,000 cells in sample one, they're B cells. I aggregate all the 4,000 cells into sample one B cells. So if you choose to attribute, what we're going to do is we're using the interaction term. In this case, I have 12 samples times 4 cell types. Using this method, I will have 48 samples, pseudo bulk samples. Okay. So now, again, if I use a raw count, I like to use the sum. If you use normalized count, you might want to use mean, and then you click finish. After you've done that, you can see the circle compared to this. When you see there inside there's a cell, means that this is single cell data. When you mouse over, it will tell you how many cells. 12 samples of 57,000 cells. But this one is 48 pseudo bulk samples, and the same number for genes for every gene. You can perform downstream analysis, but I would highly recommend you don't do that because your analysis is already really complicated. And we have this option. Any circle, when you click on it, you have a create project from data node option. So you click on that, and that will create a new project. And I'm going to just go back to the new project. This is my 48 samples. So basically for each cell type in each sample, I will consider it as a sample. And the attribute will put it over, but the name might not be very informative. You can just change the name. We just call that, attribute 1, attribute 2. I just clean it up, for every sample, I know old versus young, and also the cell type, I know, B cell and whatever, and then the gender is still there. So now this data node is 48 samples, it's still 7,000 genes. And you can, since this is raw count, think about the bulk RNA-seq. You almost treat it as a bulk RNA-seq. And if you want to use the DE-seq for differential analysis, and you can, use the DE-seq normalization to normalize the data, perform a differential analysis. Let's take a look at the PCA of this bulk level data. And now each dot is a pseudo bulk, and we can color based on, right now we color based on age. I don't see there is a whole lot of difference on age, but I'm kind of see some groups over here. Now, if I want to use the color to represent the cell type, now it makes sense, right? So cell type is the biggest variation. So that's the whole point you want to do single cell analysis, because different cell type, they are very different, the biggest source of variation. And over here, monocyte is very different. It's separate the most on the first PC, and all the others kind of on the second PC separation, right? Also, maybe I can use the size to represent age, and I can use the shape to represent gender. Now with all the different perspective, and you can look at the data, and all the downstream analysis, you can compare the genes showing differential expression between the cell type, or between the age, or between gender. You can add all your attributes to configure your differential analysis model. Okay, so that's the one other perspective for you to look at your single cell data analysis. Okay, with that, I'm gonna just wrap up the single cell RNA-seq from multiple samples analysis. We talk about how to visualize the data each individually, classify the cells individually, and combine them together to visualize them. Not only you can detect the differential expression gene, we can also look at the different cell number between your phenotype. And also we talk about the automatic cell type of classification and a pseudobulk analysis. So next, I want to show you how to analyze the SciSeq data, what options we have in Partek flow. SciSeq is for each cell, not only you capture the RNA, you also capture the cell surface protein. So we have another dimension. It's kind of multimodal, another dimension to look at just each single cell. So when you use a 10x H5 file, the H5 file, once you import, this is a 10K PBMC data, they containing both, like this is 33, 5, 31 features containing both gene expression and RNA. The import is still the same, normalization, the cleanup, filtering, filter cells, and filter features very much the same. So you have the option to do a split by feature type, because in this 10x H5 with sparse matrix output, they tag gene and protein using different tag, and based on that, we can separate them. When you click on this split by feature type, we'll know one tag is called the antibody capture, the other is called the gene expression. So this will give you the option for you to look at each type of features separately. You can use your protein to perform UMAP or t-SNE, and you can use the gene expression to perform t-SNE UMAP cluster and to see how consistent they are. So the analysis is going to be very much the same. And also we have the function. Usually you want to normalize them differently. And for protein, in this field, people use the CLR to normalize, but gene expression, you use a different one. So you can use the same normalization or you can use a different normalization. But after you normalize, if you decide, hey, I want to combine them back together, click on any of the data nodes. And in the pre-analysis tools, we have the merge matrices. So you can merge them together. You can merge cells, or you can merge features. In this case, we're going to merge features. And Partek will say, oh, okay, this you can choose which one you want to use to merge. So now, as you can see, this this green one is I merged the matrix back. And after you merge, you can also, on the merged data, you perform another level of visualization and differential analysis, right? Or another way is I want to merge only for visualization, because the protein, if you look at the protein, there's only 17 proteins, and gene expression there, 10,000 genes. When you merge them together, your visualization, your PCA or UMAP will probably be dominated by your gene expression, not protein. So there is a way called, after you've done PCA, there is a method called weighted nearest neighbor. We call it a find multimodal neighbors. Basically, if you want to integrate different assays or ataxic RNA or protein RNA at PCA level, you can use the weighted nearest neighbor. So use this function to combine them. It's kind of a merge, but at the merge, they calculate the weight. And then after merge, you can visualize, perform the UMAP and t-SNE, etc. So once you have all the different visualization, you can put them in all one graph. Here's what I saved. I have the UMAP. You can see some information is captured in one assay, but not the other. So this is the UMAP based on protein only. This is based on gene expression only. This is the UMAP based on the WN using weighted nearest neighbor. As what we can see here, for example, if you look at, I already colored them, CD4 positive T cell and CD8 positive T cell. So if you only look at RNA, they really much cluster together, right? They don't separate very well. But if you look at the protein can separate CD8 and CD4 very nicely, and even have more subtype. So some information is captured by one, but not the other. They kind of complement to each other. And if you use WNN, they will separate that pretty nicely. Okay, so again, different perspective to look at the data. Also, when you have a size-seq, you can look at, this is an example of the CD4 protein and the gene expression. As you can see, the CD4 gene expression on the x-axis, they are all in the log space. But on the x-axis, those group of the cells, the gene expression of CD4, they are 0, right? But for protein, they are not. So which means they probably are CD4 cells, but gene expression, because the dropout, you didn't capture that. Using cell surface protein CD4, you can capture those cells. You can identify that they are CD4 T cells, right? Okay, so that is size-seq. All the other analysis is very much the same as RNA-seq. It just gives you more perspective to look at the data. The other example I want to show you is the spatial transcriptome. In my understanding is the spatial data is also give you the morphology context to help you identify your cell type. And it's because the t-SNE UMAP, they all dimension reduction. When you map like a 10,000 dimension space to 3D, you're going to lose a lot of information. No matter what you look at, you only look at one tiny percentage of the whole information, right? That's why we have to use, gene expression value, use the statistics, using all the different techniques to combine them to look at your data. But transcriptome technology provides the real morphology information, and you can really see, okay, where the layer of the cells. I don't have time to show you the import, but it should be very straightforward. You just follow the wizard to import. They will give you, for the spatial data, you're going to have a gene expression, and also there is some spatial information, like the location of the spot. In Cell Ranger, they will give you a folder. You just zip up the folder, upload both together to import the data. I'm going to quickly then show you how you're going to utilize the spatial information. For the transcriptome information, you can use the same technology as we learned before. But on top of that, let's add spatial information. I will get the data. Once you import the data, we'll have the spatial information. It's only 2D. And the spatial information, they will have the X, Y location of the slides. And that's the spatial image for the data. And within that, at the bottom, you will have the background. And for the VCM data, they will have a different level of image. They have a lower resolution, high resolution, and they have aligned differential image. You can use a different image to look at your data. And you can choose not to show the background. This is the only look at the transcriptome spot. Okay, so that's the operation you will have for the spatial data. And also, if you go to the style, and the opacity will help you to look at the more histology image, and then you change that. So on top of them is your transcriptome information. So if you want to combine with the spatial, if the tissue information is more important, I can make the point size a little bit smaller, right? And you can zoom in and make it a little bigger if you want to do that. And you're going to use the same in technology we have, for example, just to quickly show you, this is a sample 1. And then if you go to, because of the slides, you can only look at the one sample at a time. Sorry, it's under axis. You go to sample 2. This is a mouse brain tissue, posterior and anterior tissue. Okay. And also you can combine with your, what we learned before. Suppose I want to add my UMAP information. Okay. UMAP. I want to look at the split sample. And I want to add the graph-based information. Graph-based cluster. I can color over here. Sometimes when you color, over here, and you might not be able to identify your cell type, but if I color on the histology image, you probably say, oh yeah, I do see like once you have a transcriptome data, UMAP is not that useful anymore. I think most people, they will focus on the histology information. You can utilize, both. I would get my biomarker table. Suppose I found some genes that this is anterior, and this, and make sure your biomarker table, they are consistent with this posterior. So suppose I can see, this gene information. Let me just duplicate. I like to duplicate it. This is using statistics to color my tissue type. I think it kind of makes sense. And now I'm going to overlay the biology to color. And then you can, if you see some familiar genes, you just color them to look at it. And another nice thing is if you want to select the cells, suppose, you use you can zoom in and make the dot a little bit bigger. Okay. Suppose you want to select some cells, over here. We have a new selection. It's called the paint mode, and only in this new version. I'm not sure whether on NIH the service updated. I already sent the information to your IT, and they should update it pretty quick soon, if not have updated yet. So you use this paint mode, you can just select. As you go along, you select all the cells, right? So whatever you want to select, you can change make it bigger, to select more as you go along, or you can make it smaller. Okay, so that's another new feature in this new version of Flow. The concept of identify the cell type is going to be very much the same, and using spatial transcriptome, you will have one more layer of the information to look at your data. With that, I would like to take questions.